So for chapter 3, we're going to start talking about the cells. And this is going to be part 1 of 3 videos. So the chapter itself is actually broken into 3 sections. And we're going to cover chapter 1. There's another video for chapter 2, and then a second video for part 2, and then a third video for part 3. So the chapter is kind of broken down that way. This beginning part is going to talk about just a general overview of cells before we go into part 1. So when we're looking at cells, just a little information, some background information to make sure we're all talking about the same things. When we're talking about a cell is the structural and functional unit of all living things. So remember, with A&P, we're talking about structure and function a lot, how the structure of something allows it to have its specific function. So cells are the foundational premise of how they are organized, allows them to do their specific things. So we've already focused on the chemistry and on the organ, more on the foundational level. And now we're going to start with cells. So they're the structural and functional unit of all living things. How they are actually organized and then the entire organism depends on this actual thing. And remember, this whole complementary principle is that the function of the cell is dictated by the shape of what the actual shell is like and all the structures of the organelles inside. So cells, there's actually a ton of different kinds of cells. There's over 200 different types of human cells. We're going to talk about a bunch of different ones in this semester. You'll talk about even more in A&P 2, but this particular chapter is going to go through the general cell, and then as we go, we'll talk about the cells for each specific system with chapters that are coming up. So when we're looking at cells, like I said, there's over 200 different types of cells. There are some that are going to be round, like an erythrocyte was a blood vessel. Some are going to look very weird, like this nerve cell over here. And some are going to be long and skinny, like a skeletal muscle cell. But they all have different functions, so they all look like different things. They all have different structures. So in this first part, we're going to talk about the generalized cell. So the generalized cell is also called a composite cell. And essentially, this is just going to be all of the main parts of a cell are going to be put together into the stereotypical general cell. And then as we go through later chapters, we'll talk about specifics that break these rules or add to these structures. So there's 3 basic parts to the generalized cell. And this is the way the chapter is actually going to be broken up. So there are 3 basic cell parts. The first part is going to be the plasma membrane. This is the heaviest part of this chapter. I'm going to go ahead and give you a heads up. This is very how things work, the physiology. There's going to be some chemistry, there's some physics happening. The plasma membrane is going to be pretty involved. The cytoplasm is the second basic part for this particular class. We're not going to focus a lot on the cytoplasm or the organelles, but I have uploaded a few helpful tricks and tips of the trade to kind of go through and review those sections, but lab covers this a little bit more in depth. And then the third part that we'll focus on is going to be the nucleus and where you'll find the DNA. So this is the way the chapter is broken up. Today we're going to focus on plasma membrane, part 1. So this is the generalized structure of a cell. So when we're talking about the nucleus, we're going to have the cytoplasm, and then essentially this outside border all the way around is going to be your plasma membrane. So that's the general view. You'll see this structure image a lot. So when we're talking about a cell, we're talking about intracellular. So here's my generic cell. You're going to have extracellular. Extra means outside. And then you're going to have intracellular, things that are inside. So just make sure you know these terms. Extra is going to be outside the cell, and intracellular is going to be inside the cell. So things that you're going to find inside the cell are going to be all of these items up here. But you will find some things outside of the cell, and these are going to be these items here. So you'll have fluid, because a cell is never just an isolated structure. It's always going to be in some sort of fluid. So outside of the cell, there's always going to be these extracellular fluids, usually the intracellular fluid, which the cell is actually going to be in. There may be fluid that's blood plasma or cerebrospinal fluid, but that extracellular fluid will always be the fluid that the cells are found inside. Okay, so to focus on the part one, this is pretty hefty. It's going to start with just a bunch of concepts, some terms, making sure we're all talking about the same thing, and then it's going to go into some nitty-gritty physiology at the end of it. So part one, we're focusing on the plasma membrane. So when we're talking about the plasma membrane, we are talking about the barrier between out and in. So it is the barrier between the internal environment and the external environment. So it will separate the outside versus the inside. And the way it will actually do that is, what it's really focusing on is, here's my barrier. You will have fluid on the inside that is going to be called intracellular fluid, and then you will have that fluid on the outside, which is the extracellular fluid. And these two fluids are what you are basically trying to separate, because they maintain the integrity of the outside environment versus the integrity of the inside environment. This plasma membrane, you'll also see the term as cell membrane. They are both the same in human physiological systems. A cell wall may be a term you've heard before. Cell wall is specific to plants. So in the human body, in living organisms, it's going to be the cell membrane or the plasma membrane. So it is going to consist of lipids. So these membrane lipids, remember we talked about these in chapter 2. Lipids are fats. They're going to have a very specific structure. And these membrane lipids are going to have a flexible lipid bilayer. And within this flexible lipid bilayer, we're going to talk about what that looks like. Another thing that this is also going to have is membrane proteins. So we're going to talk through those. What type of membrane proteins? Remember, we also talked about these in chapter 2. And then there's also going to be surface sugars. So this is also going to be called our membrane carbohydrates or our membrane sugars. So those three types of molecules we talked about in chapter 2 are coming back up immediately: our lipids, our proteins, and our sugars. Cool. So when you look at this, this is going to be what the plasma membrane is kind of described as, and it will be called a fluid mosaic model is what it's called. And a fluid mosaic model means that when you look at this plasma membrane, I'm going to draw on top of it, you'll notice that this guy right here, here is my plasma membrane. He is not a solid structure. This plasma membrane has the ability to essentially be able to not be a solid structure, but to have everything float inside. So this is going to be my plasma membrane, and then inside of it, I'm going to have, here is a protein. Here are some of my sugars. These guys are made up of lipids. So all of these are not a solid, hard structure, this plasma membrane. It's fluid. So all of the items or all of the molecules, specifically the protein ones, will float inside this plasma membrane. So it has the ability to move. It's not a solid structure. Think about more of it as like a jello, kind of like jiggly. They have the ability to float and move around. Okay, switching and looking at our first section, we're going to have our membrane lipids. What makes up our plasma membrane is going to be some lipids. Our fats. So this is a lipid bilayer. So when you talk about the plasma membrane, you will also hear the term phospholipid bilayer. So the plasma membrane is considered a phospholipid bilayer. So it is made up of phospholipids, and bilayer means it is 2 layers. So we've got our phospholipids as the base, and it is going to be 2 layers. So there are 2 parts. Remember, flashing back what a phospholipid looks like. So chapter 2, in a phospholipid, it's a modified triglyceride, which means it has a glycerol backbone and then a fatty acid tail. So this with an attached phosphate group. So this is a phospholipid. So with this, we said that with this phospholipid modified triglyceride, one part of it would like water, one part of it would not like water. So when you look at this phospholipid right here, this phosphate head is going to be water loving. It really, really likes water, which means it is going to be hydrophilic. This tail right here is water hating. So it is called hydrophobic. And this is a bilayer. So what actually happens when you have this phosphate head, which is hydrophilic, and the fatty acid tail, which is called hydrophobic, what you'll get is you will have, this is kind of a generic phosphate head, phosphate, and the glycerol tail. Fatty acid tail. So this is going to be a phospholipid. When you put them in together in a layer, like this, one layer, this part likes water, this part hates water. But remember, this whole thing is to separate the inside from the outside environment. So if I have water on the outside of my cell, it's going to love my phosphate heads. So I'll look like this. But also remember, you're going to have inside H2O because it's your intracellular fluid, and they like water. So here's my phosphate heads. They like water, but my tails run far, far away, so it doesn't like water, so it looks like this. So the way you get this phospholipid bilayer is because the phosphate heads will be facing out of the cell where the extracellular liquid is, but then the phosphate heads will be facing inside the cell where the intracellular fluid is. And the tails that does not like water will orient themselves inside these two heads. So you will essentially get a phospholipid bilayer, layer 1, layer 2, and that will look like this. So I'm going to jump up to look at this guy right here. So if you look at this phospholipid bilayer, you can see how here is my phosphate head, here are my tails, here is my phosphate head, here are my tails. And you create a layer all the way facing the external cellular or the extracellular fluid, and then you create a layer facing the intracellular fluid. So the membrane lipids are really, really important, and it's made up of 75% of this lipid is going to be this bilayer. So it's really cool how that works. There's a couple other lipids that will come into play, not just these phospholipids, which are 75%. You'll also have glycolipids, and you'll also have cholesterol, which are going to make up this cell. So then you're also going to have membrane proteins. These membrane proteins are going to live in this phospholipid bilayer. So if our phospholipid bilayer is actually making up the wall itself right here, our proteins are going to be embedded inside this phospholipid bilayer. So what are our kinds of proteins? The reason we have them, before we get into the different kinds, is that these will allow for communication with different cells and across the environment. They all are going to have specialized functions. They're very, very important. Some are going to float in the middle of the cell plasma membrane, and some are going to be attached to the actual structures inside the cell. So these are described by the two types. You've got integral, and you will have peripheral proteins. But remember, these are responsible for communication with the environment. That is why we have these. We have to somehow get communication inside and outside of the cell. So there's two types. Your integral proteins. When you have an integral protein, this type is going to be firmly planted in the plasma membrane. And I'm just going to shorthand that as PM. Firmly planted in the plasma membrane. With it being firmly planted, most are transmembrane, which means they will span from either side. So if we were drawing our plasma membrane, and I'm just going to do it as a highlighter to kind of this is one layer of our plasma membrane, This is 2 layers. So there's our phospholipid bilayer. When we say an integral protein, and most are transmembrane that cross to either side, they span the sides of the plasma membrane like this. So these will span both sides. We're going to talk about a different protein that doesn't span, which will look like this. So it does not span both sides, but a transmembrane protein will span both sides, and it looks like that. When we're talking about these, they are going to have both a hydrophobic and a hydrophilic region, and they need that because the middle part right here has to be hydrophobic because it hates the water. So this part will be hydrophobic, this part in the middle, and then these parts right here will be hydrophilic, and it has to have that so that way you know that the hydrophilic part likes the intracellular fluid that has water, and it likes to talk to the extracellular fluid that has water. So those are really, really important. And these can have a bunch of different functions that we're going to talk about. Okay, the second type of protein is going to be peripheral proteins. And a peripheral protein is loosely attached. It is not firmly planted. And these are usually things that are not going to be crossing the whole, both sides of the plasma membrane. So these are things that can be removed. They are loosely attached and they can be removed. These will have a bunch of different functions, and we're going to talk about these as well. So here are the functions of our plasma membrane proteins. So functions of our, I'm going to shorthand, plasma membrane proteins. These are the 6 we're going to talk about. The functions are transport, receptors for cellular transduction, attachment, enzymes, and intracellular joining, and then cell-to-cell recognition. So these are all on individual slides, so we're just going to talk about these. So transport is the first function of a plasma membrane protein, and so it'll actually allow things to move across the membrane. So it acts as the bridge. It spans both sides, and it allows things to be able to move from outside the cell to inside the cell. Let me find a color. There we go. So it will move these things from out to in, or it will move things from in to out. It transports across the plasma membrane. So it's the bridge. We'll talk a lot about this in depth as we go later on in this course. The second function of these plasma membrane proteins is going to be receptors for signals. So this is when we've got something that is traveling inside our body or inside, usually going to be inside our blood. These receptors are going to actually have a specific binding site that fits the shape of some other kind of chemical, which is usually going to be kind of like a hormone. And when that hormone happens, it will bind, it will change the shape of the protein, and then it will initiate a change of chemical reactions in the cell. So it's going to be a signal. So if this is our cell, and this is our outside, our extracellular fluid, and this is our inside, and we have our protein, when this receptor binds to it, it will essentially cause some sort of chain of events inside the cell. So it doesn't cross the cell, it does not actually cross across the plasma membrane, but it essentially gives the go signal to cause things inside the cell to happen. So it creates that signal for go. And that's really important for a lot of physiological processes. The third thing is it's going to be an attachment. They are an anchor. So this guy right here spans the plasma membrane. So he spans the plasma membrane, and you can see he's got anchors on this side and he's got anchors on this side. So he will act as a cytoskeleton, and he will help anchor proteins and also other movements inside the cell. So he's an attachment. He's going to act as an anchor. Another type of protein function is going to be an enzyme. These membranes, proteins can act as an enzyme. So this was covered in chapter 2. or an enzyme can allow the chemical reaction to occur without getting used up. So these can be the enzyme that is the active site. So these can actually act as the place that those chemical reactions will occur. So here's my chemical reaction. I had a circle go in, I get a square out, but my enzyme was the actual place where that occurred. So these enzymes can live in your plasma membrane. If you're unfamiliar with the enzyme is, go back and review that end part of chapter 2. So if that was number 4, the 5th and 6th part is going to be cell-to-cell joining. So they can connect other cells. So what they can do is they can connect adjacent cells and they can hook them together. And the way they do that is they have these things called cell adhesion molecules, or CAMs, which are super sticky cells. So a cell adhesion molecule acts like tape. And it will take a plasma membrane. Here's one plasma membrane. Here's my second plasma membrane. And it will take my protein from here, and it will take my protein from here, and it will glue them together. So that way my cells are now attached. The sixth function of a membrane protein is going to be cell-to-cell recognition. So number 5 was cell-to-cell joining, where we are connecting cells. So we're just simply gluing them together. In cell-to-cell recognition, what we're doing is it's going to have an identifier. So essentially it acts as the license or the facial recognition of the cells. So if I have my plasma membrane of one cell and my plasma membrane of another cell, and I've got my proteins like this protein right here can act as my identifier. So this guy, he will talk to this guy. This is really, really, really important in immune response. So this is how you actually know that if you've ever heard of someone who has an organ transplant and their organs reject some of these other organs, some of this is coming into play where the proteins on the cell that is natural to their body does not recognize the proteins that have been added to their body, and they reject them, essentially turn them away because they do not recognize them. So this is a very, very, very important response, especially in immune response, but it's cell-to-cell recognition. Okay, so that was membrane proteins. We did membrane lipids, and then this glycocalyx is your membrane sticky carbohydrates. So this is going to be your membrane sugar. So these 3 things make up the foundation of your plasma membrane. These are made up of carbohydrates that stick on the outside of the plasma membrane. So they will stick out onto the cell. Some are going to be attached to lipids. If it's attached to a lipid, it's going to be a glycolipid. If it's attached to a protein, it's going to be a glycoprotein. So remember, when you're looking at those, the prefix in science is really, really important. So the prefix here is glyco, which means sugar. So if it is sugar plus a lipid, it is a glycolipid. If it is a sugar plus a protein, it's a glycoprotein. So it's either a sticky or sugary lipid or a sugary protein. But these are really important, and they can also act as these 2 cell recognition. This is what they're really, really important for. So if I were to draw, I'm going to go up to that general composite cell that we looked at so we can look at all of these pieces. So when you're looking at this plasma membrane cell right here, here is our plasma membrane right here. You can see him, one layer. Here is my second layer. So all the way around is my plasma membrane. So my plasma membrane, layer 1, plasma membrane, layer 2. Here is my protein. Here is another protein. These are transmembrane proteins, so they span the cell. Here is my glycoprotein. See that sticky sugar? That is cholesterol. That is sticking out the top of my protein. That makes it a glycoprotein. Here is my sugar. He is attached to a glycolipid, so that makes him a glycolipid. So you can see how you have your phospholipids, your proteins, and then your sugars, and that makes up the plasma membrane. So use that to go back and understand how all these things work. So moving on. These are really, really important. So the sugar protein or the glycocalyx of the cancer cells are what help with understanding whether or not the immune system can recognize if the cell has been damaged or not. And some of the mutated cells, they don't have the ability to replicate. So it's important to know which cells to get rid of, and that glycocalyx is responsible for that. Okay, so we've talked about the individual cell plasma membrane and what makes it up, but what happens if you need to connect cells? And this is called cell junctions. We are not isolated cells, so I like to describe cells as Legos. And if we are going to put all of our Legos together, you don't really have any function if you just have one individual Lego. So all of our Legos need to connect and create larger organisms. So these are going to be cell-to-cell junctions. Our cells are not free. They are always bound to other cells. Some cells are, but these are going to be blood or sperm cells, but the actual structural cellular unit is not going to be free. They are all bound together to form tissues and organs. And there are 3 ways they can be bound together, and we're going to talk through these guys. So 3 types of cell junctions. The first type is going to be a tight junction. How these cells connect is called a tight junction. With this particular cell, what it is integral proteins of adjacent cells form to make an impermeable junction. It is an impermeable junction, and it prevents fluids and most molecules. So when we are talking about a tight junction, these are going to be integral proteins that are sewed together. So if they are sewed together, they are impermeable. They are fused. And it is important to note that it is an impermeable junction. Impermeable means not even water can get through. So no liquids. It is a waterproof seal. So an example of this is going to be in your digestive tract. You do not want, or in your skin, you do not want to have, well, one layer of your skin, but you do not want to have a liquid be able to pass through. So this example right here will show here is one cell. Here is my other cell, and he is connected to a third cell. The way these are connected is these are actually going to be sewn together. So if you look right here, you can see that these guys are sewed together. The way they are connected is nothing is able to get through these cells. So they are sewed together and it is impermeable. You can see this right here, how they are sewed together. It's very, very cool. Another type of connection is going to be desmosomes. And these desmosomes are going to be rivet-like cells. So they're going to be binding bodies. They are mechanical couplings. So there's actually an additional structure that keeps these guys together. And these are going to kind of be like a zipper, kind of like a rivet. So these desmosomes, if I have my cell right here, and I have another cell right here, they are going to connect because it will go from one side of this guy and one side of this guy. They're going to be a mechanical coupling. They're going to adhere together. These are going to be called catecherins. So this guy that connects them is called a catecherin. And essentially it acts like a screw, if that's the easiest way to describe it. A catecherin is a screw. So these will lock together, kind of like the teeth of a zipper or a bunch of different screws. However, as you know, if you were to put a bunch of screws into something that doesn't have the ability to maintain it, it'll break through. So what you also have is not only do you have these catecherins, you are going to have this thing called a plaque. And the plaque is going to be found on either side of the plasma membrane. On the inside, it's essentially going to be a washer, if you're familiar with terminology. So this is going to be your plaque. And it is a tough, hardened lining that is going to line the inside of the cell. And then you're going to have your cadherin that will be crossing over top of that. So this plaque acts as the washer, where the cadherin acts like the screw, and it will anchor these two cells together. So you need to know about the plaques on the inside of the plasma membrane, and the cadherin are the linker proteins. So those are really, really important. And then the other thing is there are going to be some keratin filaments inside the cell for added strength. So that means inside this cell, there is going to be some proteins that are holding this guy together so he doesn't pull apart. These are going to be your keratin filaments. They're added strength. So your desmosomes are going to be screwed together. So here's your cadherin, here's your plaque, and here's your keratin filaments. That way you can see how the whole thing works. So that's a desmosome. What's important for a desmosome is it will still be able to fit these cells together, really, really important. And it works really well to bind cells into sheets. So it's going to be more of a long connected piece. So your skin will be connected through some of these desmosomes. So that's really important when we get to that chapter. The third type of connection is going to be called gap junctions. And gap junctions are going to form by little tiny tunnels called connexions, and they will allow molecules to pass from cell to cell. So it also allows electrical signals to pass quickly from one cell to the next. And the way that works is, here is my one cell, here's my other cell, but it's a gap junction, it's a tunnel. So rather than being separate, my eraser erased the whole thing. So rather than being separate, only, whoa, so rather than being separate, it's just this tiny little space between the two that's missing, and it forms a bridge. And this bridge allows for anything in here to be able to cross over and go through here. So it creates this little tunnel, which is your connexion. And it allows things to travel really, really quickly because nothing has to pass over a wall. It is just connected liquid to liquid. So down here, you can see how we've got these channels, these little proteins that will connect this liquid from one cell to the other. So how do we actually move things across the plasma membrane? So movement of substances across the plasma membrane is very important because things need to move. Here's my cell. Things will need to move from outside the cell into my cell, and things will need to move into my cell and out. So we have to be able to move things across this plasma membrane, but everything that we've just talked about, it is a very specific structure. So your plasma membrane has something that is selective permeable. And selective permeable means that some molecules pass through, but some molecules don't. So how do we get things out, or how do we get things in if some things are not allowed to actually pass through this plasma membrane, but some things are? So that selective permeable is really important because we don't want dangerous things to come into the cell, but we also want things that we need to get rid of to get out of the cell. So it's kind of a catch-22 protection, but we also have to work around it. So there's two ways that we will actually have substances cross the membrane. Passive, no energy, and active, it will require energy. So we're going to talk through passive transport first. So section 3.3, we're going to talk about passive transport. So passive transport across the plasma membrane is no ATP, no energy. There are two types. You will have diffusion, and there's two types of diffusion, or three types of diffusion, and then you will have filtration, we're going to spend most of our time talking about diffusion. Filtration will come mainly when you get to the urinary system or to the cardiovascular system, which is going to be A2. So this class will spend most of our time talking about diffusion. So what is diffusion? This is a form of passive transport. So we're moving things across the plasma membrane. And it is going to happen because we are going to move molecules from an area of high concentration to an area of low concentration. So this is going to be called a concentration gradient. So with diffusion, we are following something called a concentration gradient. And anywhere you have a high amount of something, by definition, if there's a lot of something here, somewhere else there will be low something. We are talking about a concentration, so it will be done in brackets. So if you have a high concentration and a low concentration, things are always going to move from high to low. So with a concentration gradient, it will be the diffusion of molecules from an area of high concentration to an area of low concentration. That is the foundational premise of this whole section right here. So this is super, super important. It will always go from high to low. Super, super important. Always go high to low. It is not right or left, and it is not up and down. It is always going to be high to low. This is really, really, really important. To do this, as a reminder, energy is not required. So this is happening just because of the nature of our kinetic energy and these molecules hitting each other. So diffusion is high to low, and it is very, very, very important. And the speed is affected by both the size of the molecule and the temperature. Remember, the larger the molecule, think all the way back to chapter 2 when we talked about energy, the larger the molecule, the slower it moves, whereas the faster the temperature, the higher the temperature, the faster it moves. So here's an example of you put some dye in, and it'll essentially diffuse until the whole thing is distributed. No energy was required. So this naturally happens. They want to do this. Molecules want to move, but the plasma membrane will stop and create this concentration gradient. So if things are always moving and it's always going from high to low, your plasma membrane is acting as the barrier, and so they're not able to do this. So we have to be able to somehow get it across to be able to move from high to low. So if your plasma membrane is severely damaged and you don't have this barrier and you just have things moving, you have a lot of physiological issues. So for example, burn patients who do not have their cell wall or do not have their skin also have, that's an example of if they don't have their skin, they're not connected, there's not a barrier between their inside body and their outside body. Think about that on a smaller scale of the cell. If there's no cell wall, there's no connection and separation from the inside of the cell to the outside of the cell, and that's very, very, very important. Okay, with this diffusion, we're going to focus on the phospholipid bilayer, because with this, what we're really focusing on the fact is that one side of this really likes things. This is how it's produced. One side does not. So this side really, really likes water. That's the phospholipid head. This side really, really likes water. But this section in the middle hates water. So things that are dissolvable in water can't pass through the plasma membrane super easily because they can't pass through the area that hates water. So for things that are dissolvable in water, are not necessarily able to pass through. Items that are super large are not able to pass through. So this is a very, very, very important. So some things that will passively move through are going to be lipid soluble. So things that are going to be like lipids. So it's going to be not the things that likes water. Those will typically pass through. And then very, very small molecules can typically easily pass through. It's all of the other items that we need help with. So How we're going to do this is the first type of diffusion is going to be simple diffusion. With simple diffusion, these are going to be focused on lipid soluble, so things that will dissolve in lipids, also known as nonpolar. All of these are hydrophobic, so all of these do not like water, but these can all pass through the bilayer. So all of these have no problem passing through the phospholipid bilayer. So simple diffusion, things just pass through. Some examples of these are going to be oxygen, carbon dioxide, and fat-soluble vitamins. And here is your example. They like the hydrophobic inside, so they have no problem with this section. So they simply pass on through. Simple, simple, simple. They move from an area of high to an area of low. Another type of diffusion you can have is facilitated diffusion. And with facilitated diffusion, this type, you are still moving things from high to low, and you are still moving items through the plasma membrane, and this is going to be certain hydrophobic things, but they are going to have to be transported passively down their concentration as well by some sort of mediation. So there's going to be some sort of mediated, facilitated diffusion. And basically what this means is that there's going to be help crossing the plasma membrane. Again, plasma membrane. So this facilitated diffusion, they still passively move from point A to point B. They move down their concentration gradient, but they just have a little bit of help. So the first type is going to be carrier. The second type is going to be channel. So when we have a carrier-mediated facilitated diffusion, that is a very long term, but what I want you to know is you know what diffusion is. So things are moving from high to low, no energy. If it's facilitated, it needs help. And what is happening is there's a carrier. So a carrier is a transmembrane integral protein. So these are going to like specific things such as sugars or amino acids that are too large for the membrane channel. So the way this is going to work is here's your little image down here. Here is my carrier protein. So a carrier-mediated facilitated diffusion is you're going to have a carrier protein, which is going to essentially transport the molecule across. It's like a raft or a stepping stone. It exists. It essentially gives that in-between step to be able to default and move around that hydrophobic inside of the plasma membrane that these items don't like or are too big. So it essentially acts as a stepping stone, and it's able to help the molecule get across. So it's a carrier. It's facilitated diffusion. The other type is channel mediated facilitated diffusion. So it's the same thing. Remember, diffusion, so you know you're moving things from high to low. It's facilitated, so it's going to have some sort of help. And the way it's helped is there's going to be a channel. So these channels are going to essentially be the bridge. So the other one was a life raft that you had to kind of get in to float across. You had to be a part of it. Whereas the channels is essentially going to be a hollow core. These are going to be proteins, remember? So it's a protein with a hollow core. So down here in my example, here is my plasma membrane, here is my channel protein, but you can see these guys can move straight through down this plasma membrane. So that is how this guy works. It's just a straight channel or bridge to be able to get through. There are two types of these guys. So like any bridge, if you're going to have a plasma membrane, and it's going to have a protein that goes across, and this protein is going to be able to allow things to come into the cell. So it kind of looks like that. So here is my protein, and there's a channel now that things can come in. these are going to have some sort of gate. So there are two types of gates that will keep this bridge open. There's going to be a leakage channel, and a leakage channel has no gate. It is always open. So that's just a bridge that is always open. But then you're going to have a gated channel, and a gated channel will have some sort of block, and it will only open for some sort of chemical or electrical signal. So it has to have some sort of signal to open up the gate so that things can come in. These are going to come into play a little bit later in this section. So in the diffusion, we are moving the item, we're moving the molecule across the plasma membrane. But there's also a process called osmosis. And osmosis is going to be the movement of the solvent. So everything we've talked about so far in diffusion, it is the solute that is moving. So it's the item, it's the particle, the solute is moving. In osmosis, it's the solvent. In physiology, it is going to be usually water. But it could be something else, but it's usually going to be water. So water will pass through the plasma membrane. So this is the movement of water. Water will dissolve through the plasma membrane. So the flow of water occurs when there is a different concentration of water on either side. So like we said earlier, you can have an area of high concentration and an area of low concentration. Water works the same way. Water will move from high to low. So here we've got our osmosis, so you can see this thing. However, one thing that's interesting to note is that water and solute are inversely related. So if we look at this little example right here, and let's say we are outside the cell, and now we're inside the cell, and we're going to put our phospholipid bilayer down the middle. So we need to be able to get things across the cell. So here is our example. Actually, I am going to match the picture, so we are all talking about the same thing. So I'm going to take this image right here. Here is my plasma membrane. This is going to be outside the cell. This is going to be inside. As a heads up, whenever you see the book and it's blue, it's out, and whenever you see yellow, it's inside. So out would be the extracellular fluid, the in would be the intracellular fluid. So this is the extracellular fluid, this is the intracellular fluid. So what we're doing is we're trying to move both water and solute. So we're going to make this a sodium ion. We have water on both sides, and we have a solute on both sides. However, when we look at it this way, typically, if you look at it right here, there's 1, 2, 3, 4, 5, 6, 7, 8. There's 8 water molecules on that side, and 1, 2, 3 water molecules on this side. So you can have a different amount of molecules on either side. So the osmolarity is the measure of the concentration of solute. It's how many molecules there are on either side. So osmolarity will be the solute particles. It's the solute concentration. The solute concentration will be on the outside and on the inside, but you'll also have water on the inside and on the outside. So there's going to be a chemical equation that kind of is the solute to the concentration of the solvent. How much of the sodium there is in how much of the water. So this is kind of the general equation that we're going to talk about. So whenever you have more solute, you will have less solvent. Whenever you have more solvent, you will have less solute. So these are an inverse relationship between the solute and the solvent. Whenever there's more of one, there will be less of another. So when the solute concentration, the water goes down. And the reason this happens is because the solute particles will actually displace the water molecules. So in osmosis, Water, that's what we're talking about in osmosis. Water moves by osmosis from areas of low solute to areas of high solute. However, like we said, it will always go from high to low. This is the direction it will always go in. But this statement is weird because I want you to notice that water moves by osmosis from an area of low solute, but it is going to be an area of high, ooh, that did not work. It's an area of high water. So it's still moving from high to low. Water is moving from high to low. It still follows that same premise, but it's going to be high water, which is going to be low solute. So up here, if we have high water, we are going to have low solute. Outside the cell, high water, low solute. Then when we come down here and it says we have areas of low water, it will be an area of high solute. So if we are going to have low water, we are going to have an area of high solvent. So outside the cell, there is going to be a concentration of solute and solvent that are going to be opposite. You'll have a low or an inverse relationship between these ties, and then inside your cell you'll also have an inverse relationship with these guys. What's important is that things are moving across the plasma membrane, and they are moving from an area of high to low. So if you have a lot of solute that you need to even out, the solute will move across the plasma membrane from high to low. But the solvent will move across the plasma membrane from high to low. So they are actually moving in different directions. So with diffusion, you're moving something across the plasma membrane, but in osmosis, as a reminder, you're moving the solvent, and it is going to be the opposite direction from the solute. It's very tricky. I want you guys to know that concept. Okay, so this will keep happening until things move across the plasma membrane and hit equilibrium. So you will have movement of your solvent and or your movement of your solute until equilibrium is hit. And when you hit equilibrium, essentially what that means is it is going to be equal amounts on both sides. That is when the process of the movement will end, is equal amounts on both sides. So you can see here's an example of, on this particular side over here, we have the same amount of water. You can see the water line is here, but this has a whole bunch more molecules. So this is going to be high and this is going to be low. So in this particular case, the molecules are going to move this way to even out. So you can see the molecules have evened out. So now that the sides are equal for the amount of stuff. What happens if you have this example and you have a selective permeable membrane? Things can't get through. So the molecules can't move. The water is going to need to move. So over here we have high sugar, all those red molecules, and we have low sugar over here. Sugar cannot pass through. So we're going to have to move the water. We're going to have to do osmosis. Do you see over here how we have low sugar, but we're also going to have high water. There's a lot of water over here, but over here we have a lot of sugar, but low water. So things are going to move down its concentration gradient. If the sugar can't move, the water will move. So water will move this way. So you can see how if the water has moved this way, it is now equal on both sides for the concentration because the water has shifted and the water has moved. So this is really important because when water moves, it creates a pressure gradient. So hydrostatic and osmotic pressure are things that we're actually not going to spend a lot of time on, but I want to read through these definitions. So hydrostatic pressure is the pressure of water inside pushing on the cell membrane. So that means if you add a bunch of water, it's water pushing against this plasma membrane, whereas osmotic pressure is the tendency of water to move into the cell. So we have an area of high water and we have an area of low water. There will be a pressure gradient that is going to send the water inside the cell. So pressure will push water in. So not only do you have pressure on the outside pushing the cells out, you're also going to have pressure pushing in. So essentially, if this is your cell, you have both pressure pushing in and you have pressure pushing out. So that's how you maintain this boundary between the plasma membrane, is that the movement of the water creates a pressure in both directions to maintain the integrity of the cell. If water can go in and out of the cell, you can actually change the cell. A living cell has limits to how much water can enter it. You can have too much water leaving a cell, which will cause it to shrink. And if any kind of change in that cell volume will disrupt things occurring in the body. So what we're going to talk about with this is going to be something called tonicity. So with this tonicity, what we're talking about is the ability of a solution to change shape. It's the ability of a solution to change the shape of a cell by changing the volume of water. The tone of the cell can change. Think of the tone as the shape. So you can add or remove water, and then it changes the shape of the cell. There are three types of solutions: isotonic, hypertonic, and hypotonic. So if we were to take this cell and we're going to put him in some liquid, if he's going to be in an isotonic liquid, so an isotonic solution, it is going to have the same osmolarity inside and outside. So it will have one item in, one item out. It will have one H2O out, one H2O in. It will have the same concentration out versus in. If you have the same concentration of out versus in, you have water moving out. At the same time, you have water moving out. So if it's the same concentration, there's no shape change. Water's moving out, at the same time water's moving in. You essentially replace the water. Your cell shape does not change. However, that can be different in the other two types. So if you have a hypertonic solution, so in a hypertonic solution, you have a higher osmolarity. You have a higher osmolarity inside the cell. So with a hypertonic solution, you have a higher osmolarity inside the cell. So there is more things. There's a higher concentration out of the cell. So that means if we're doing our out, we have a higher concentration out. So it is going to be a higher concentration out. I'm saying this several times because it's the only way to really keep track of this concept. So higher concentration out. If you really want to draw it, I like to do higher concentration out. If you have a higher concentration out, you're going to have a lower concentration of water. In. So let me draw how I would do this. So sorry, y'all. Like I said, this is a very confusing process, so make sure you're tracking exactly how you're doing it. So I'm going to start and break it down one by one. In a hypertonic solution, outside the cell, you're going to have a high amount of items. So this is our stars. If you have a high amount of items out of the cell, that means inside you're going to have a low concentration of whatever items you have. Okay, that's the solute. If you have the solvent, you're going to have a low concentration of water outside the cell. So we'll have one H2O here, but then we'll have a high concentration of H2O in the cell. So we're going to have a bunch of H2O molecules. When this works in a hypertonic solution, liquid wants to move out of the cell. So things will move from a high concentration of water to a low concentration of water. So things will move this way. So it will move from in to out. In a hypertonic solution, water moves out. If water is moving out of the cell, then that means the cell is essentially dehydrating, or the cell is shrinking. So your cell will shrink, otherwise known as crenate. Your cell will shrink, otherwise known as crenate. So you will get essentially a raisin type cell. So that's what's happening in a hypertonic solution. The third one is hypotonic solution. So if we take our isotonic and we take our hypertonic, the third one is going to be the very similar type, and it will be a hypotonic solution. So in a hypotonic solution, what you're doing is you have your cell inside this liquid, and with a hypotonic solution, you are going to have a lower amount outside the cell and inside the cell, but you're going to have a low amount outside the cell. So to start with these two, start with this topic right here. Where is the high amount? Or where is the low amount? So what I would do is I would do low amount on the out of the cell, and you have a high amount on the inside of the cell. To start with figuring out these concepts, always start with where the high concentration of solutes are. Here in a hypertonic solution, the high concentration of solutes is outside the cell, while in a hypotonic solution, the high concentration of solutes is inside the cell. Okay, that's the stuff. So we're going to have a high amount in. Here we go. What about our water? These are an inverse relationship. So I'm going to have a high amount of water where I have low solute, right here. And I'm going to have a low amount of water where I have high solute, right here. So water from high to low, I'm only going to have one thing of water inside, but I have a bunch of water outside. So my water wants to move from an area of high concentration to low concentration, always wants to happen. So it wants to move out to in. So H2O will move in. When water moves in, you can actually burst this plasma membrane. So if this happens, your cell will burst. It will first get big, but if it gets too much water into the cell will burst, and this is actually going to be called lyse. So you can change the actual structure and shape of the cell by adding or removing a solvent. This is really important because if you go to the ER because you have been dehydrated, if they were to give you a full-on solid H2O, only water intravenously, you would actually have major problems because your blood itself has a bunch of solutes in it. So whatever is given in the ER in order to have dehydration, it is not pure water, because pure water will actually cause one of these systems to happen where it will cause your cell to change shape, and if the cell changes shape, it's not able to do what it needs to do. However, what is given during a visit to the ER for dehydration is called physiological saline, which is going to have water plus some concentration of solutes that matches the concentration of solutes in your blood naturally. So they're going to give you an isotonic solution, so that way nothing changes to the shape of your red blood cells. So that's very, very important when you're getting intravenous solutions, is that the tonicity of the solution matches the tonicity of the environment. So it needs to give you an isotonic solutions, because these guys, if you give them a hyper or a hypo, it will cause damage to the cells. Okay, 3.4. Membrane transport. This is going to be active transport. So everything we talked about previously was passive. This is going to be active transport. So active transport will require ATP. There are two types of active transport, and then there's going to be vesicular transport as well. The reason you need active transport is because it does require ATP, but the reason is these that it needs to move across the plasma membrane are too large. They are not lipid soluble, or they do not move down their concentration gradient. They need to move opposite their concentration gradient. So these are the things that need to be pumped across the plasma membrane. So an active transport does require a carrier protein. These are usually going to be called pumps. So there's some sort of carrier protein that will be utilized in order to pump things across the plasma membrane. So if here's my plasma membrane, this carrier protein, essentially, if this is what I need to do, it is going to pump somehow across. Something more than one thing, but most solutes are going to move against their concentration gradient. So this is really, really important because we are moving things from an area of low to an area of high. This is not natural. It is a pump. We are going against the concentration gradient. The normal concentration gradient wants to move from high to low. That is always. But with an active transport, you are pumping from low to high. So that is opposite, but it requires ATP to do this. So there's two types, primary, and then there's secondary active transport. We're going to focus on primary the most. So primary active transport. This is the one of two major processes that you need to know from part 1. We haven't even gotten to the bulk of this chapter yet. So primary active transport. This is happening because there is energy, or ATP, that is used. So it is using energy from ATP. And the energy from ATP will cause a change, or the shape of the protein to change. So it will cause the shape of the protein to change. That shape of the protein, when it is going to move things from point A to point B. So it will pump ions across the plasma membrane. Examples of these guys are going to be calcium, hydrogen, and then the sodium-potassium pump. So this is the first big takeaway from this chapter, is going to be the sodium-potassium pump. Now, as remember, sodium is this, Na+, potassium is K+. So we're going to use these terms today. So sodium-potassium pump is the most studied pump in the body. It is basically an enzyme, so we're going to talk about the sodium-potassium ATPase, and it will pump sodium out of the cell, and it will pump potassium into the cell. So this is the direction things will move. This is really, really important when we get to nerves and muscles. So we're going to talk a lot about this. So with active transport, it is acting as a sodium-potassium pump, and as the sodium-potassium works, it's going to pump sodium out of the cell and potassium back into the cell against their concentration gradient. So that is important. We're going to pump sodium out, like we said here, and we're going to pump potassium into the cell, but it is against their concentration gradient. So how that actually works is going to be this lovely little image right here. So we're going to start with part 1. So when you start with your sodium-potassium pump, what is happening in part 1 is it is going to be, let me draw my picture, blow it up. So this is going to be the sodium-potassium pump. So the first thing that's going to happen is we have our plasma membrane. And this plasma membrane is going to have a carrier protein. And this carrier protein is actually going to be open on the inside. So with this, in step 1, this is how we're going to start. We have our plasma membrane and we have a carrier protein, and this carrier protein is going to have ATP attached to it. So this is how we start. And with this sodium-potassium pump, we are going to do 3 sodium from inside the cell. So we're going to take these 3 sodiums, 1, 2, 3, and these 3 sodiums are going to bind to the inside of the protein. So these 3 sodiums are going to bind to the inside of this protein. This is going to give us step 2. So step 2 says that, remember, we're going to have our plasma membrane, but we also said that this is going to cause the shape to change. So that's the direction we're going. So now we have our plasma membrane and we have our 3 sodium on the inside. So what's happening here is when the sodium binds, it will actually cause hydrolysis of ATP. And what that means is hydrolysis of ATP. So it will go from ATP, and it will do what we did at the end of chapter 2. You will have ADP plus a phosphate. So our ATP was attached. So now what we're going to get is we're going to have our phosphate are attached, but what we have lost is ADP. We have gone through the process of hydrolysis. When this occurs, you're going to have step 3. And step 3 is this is called phosphorylation. So phosphorylation causes the protein to change shape. And what happens when this protein changes shape? It will open to the outside. So you'll notice here, it's open to the in. But now phosphorylation is going to cause it to open to the outside. So here's my plasma membrane. Now here is my protein. He is open to the out. But I had 3 sodium ions. That means I have released 3 sodium out of the cell. But I still have my phosphate attached. So what has happened in steps 1, 2, 3? I have 3 sodium ions inside the cell, and so they have bound with my receptor protein and ATP. What happens is my ATP will pop off and it will go through hydrolysis, and I'll have ATP, ADP, and a phosphate group. When this happens, that phosphorylation causes my protein to open to the other side. So it essentially shifts gears and it goes from facing the inside to facing the outside. And it will release my 3 sodium outside the cell. So that's step 3. What's happening in the next part will be step 4. At step 4, you will have 2 potassium outside will bind. So we've just released our sodium. Here's my plasma membrane. My protein looks like this. Remember, he still has phosphate attached. And now I'm going to do squares. My two potassium are binding. And when your 2 potassium binds from outside the cell, that will trigger step number 5. And step number 5, whenever you have the potassium binds, the potassium binding will cause dephosphorylation. It will cause dephosphorylation, and it will return to original shape. So dephosphorylation means we're going to lose the phosphate. So here's my plasma membrane. Here is my protein. I've got my 2 sodiums inside. Sorry, my 2 potassiums inside. And we had this phosphate attached. Well, when the two potassiums bind and it closes this up, this phosphate no longer is attached, and this phosphate will pop off. So now we have no phosphate attached. And when this phosphate pops off, essentially it will open up our potassium into the inside of our cell. So it will cause our phosphorylation to change shape, and that will be our phase or step 6. And it will bind ATP and it will release potassium to the inside. So here's my plasma membrane. Here's my protein. I have released my 2 potassiums to the inside of the cell, but I did this because I bind ATP back to that spot. So this pumping is essentially what it's doing is it is taking 3 sodiums from inside and it is pumping them across the plasma membrane. And so you're going to end up with the 3 sodiums outside. So that's how you're doing it. You're going from inside to outside. So you're creating a high sodium outside the cell. You're artificially pumping sodium outside the cell to create a high sodium environment. At the same time you're going to do that, you're then going to pump 2 potassiums that are outside the cell, and these are to get pumped so that they are going to be 2 potassiums in. So you are pumping this way. You are moving your 2 potassiums inside the cell. So you're creating a high potassium environment inside. So you are pumping 3 sodium out for every 2 potassium in. This is the sodium-potassium pump. You are pumping 3 sodium out for every 2 potassium in. And this is how you do it. Focus figure 3.1 talks through this process. Okay, that's primary active transport, and that's the sodium-potassium pump. Remember, you're changing the shape of the protein. So what's happening next in this sodium-potassium pump is all of that is active transport, and that is primary active transport. And then the second part of active transport is called secondary active transport. And secondary active transport is an interesting concept. So basically what secondary active transport is saying is that after doing primary active transport, it has created a gradient. So after primary active transport, it has created a gradient. Here is my gradient. High sodium out, high potassium in. So this is at the end of the day, I have created a gradient. So I'm going to have high sodium outside my cell, and I am going to have high potassium inside my cell. So this is going to be after the sodium-potassium pump, you've created this gradient of high sodium out and high potassium in. But with secondary active transport, essentially it's after primary active transport, things will be moved by their concentration gradient. So sodium will drag other molecules with it as it flows into the cell. So we've created this active transport gradient, high sodium out. Sodium wants to move down its concentration gradient. So sodium wants to move out to in. So when sodium moves outside to inside the cell and down its concentration gradient, it brings things with it. When it brings things with it, it can move things across the plasma membrane that it could not do before. So some of the things it likes to bring with it is going to be a sugar. So as sodium has been pumped out of the cell, so there's a high amount of sodium outside the cell, with secondary active transport, sodium will move down its concentration gradient back into the cell, and it will carry other molecules with it, usually to be sugars. This is how it's called secondary, because something else is carrying it across the membrane. So it's a very interesting premise. Okay, so then when you look at this extracellular fluid, here's primary active transport right here. You've got your ATP, you've pumped sodium out, you're pumping potassium in, but with secondary active transport, when that sodium needs to come back inside the cell, it is going to carry glucose with it. So that's an example. Okay, another type of active transport is going to be vesicular transport. Vesicular transport is going to use some sort of vesicular sac around whatever it is transporting. So it's going to package it. It's going to be the Amazon of its time. So it's going to be a vesicular sac somehow around the molecule. And this usually does require ATP. So here's a couple examples. We're not going to spend a lot of time on this one, but I want you guys to know the general idea with vesicular transport. There's two ways of vesicular transport. You can have endocytosis, and endo is to be movement into the cell. So you are going to move something into the cell, whereas exocytosis is you are going to move or transport something out of the cell. So one way or the other, you are moving items in or out, but you've packaged them in a container. So this is how you're going to package them into a container. This is the best little image to just talk about what's going on. So here is your package. This is what we're talking about for as vesicular. So this is going to be endocytosis right here. Well, it is this little package is coming around all the stuff it needs to bring in. It has packaged everything in these little vesicles. These little vesicles are now going to dump their stuff inside the cell, and then this empty Amazon package will essentially become part of the plasma membrane. Whereas if you have exocytosis, you're going to start with this little package of items, and it will go and it will fuse to the plasma membrane. So to draw this out, if this is my plasma membrane, when we're doing endocytosis, you are moving things from outside the cell to inside the cell. Your plasma membrane will bud into little tiny packages, and it will bud around all of the stuff, and it will essentially create your plasma membrane will create this little package with all of the items in it. So that was how you'll do endocytosis. When you do exocytosis, you're going to take that same plasma membrane, and if you're going to be inside the cell versus outside the cell, you're going to take your plasma membrane, and it's going to do the same thing. It will create a little bulge. And this is all the stuff that's inside. And then it will bulge, so all of the stuff is going to be in a little package now on the outside of the cell. So that's how you're going to move things in the cell through vesicular transport. It just packages them in the same plasma membrane of the plasma membrane itself. Okay, phagocytosis is an important concept because phagocytosis is going to be where it's actually going to be called cell eating. So phagocytosis is an important type of endocytosis. and it will engulf whatever it needs to engulf, and then it will eat it. So just to come on down to this image right here with phagocytosis, you can see it's got the plasma membrane that's come around. Here's the little bulb. This is the item it needs to get rid of and it needs to eat. So what it will do is it will close this up, and then it will dissect and destroy this item. So the phagocytosis is going to be combined with a lysosome, and it will actually digest and break up the item. We're going to skip pinocytosis right now, and we're going to skip receptor-mediated endocytosis. Okay, exocytosis is the same premise of things exiting the material. So it's the process where material is ejected from the cell. It's going to also be in a secretory vesicle, but we're not going to talk about snare proteins for this class right now. So 3.5, if I said the sodium-potassium pump was the first bulk of this information, the resting membrane potential is going to be the second bulk of this information. So this is where we're going to spend the last amount of time. So membrane potential. What's happening in this section is that we are going to change the electrical activity inside our cells. So when we're talking about membrane potential, there's a couple terms we need to know. So the selective permeability of the plasma membrane. The selective permeability of the plasma membrane, letting things in and letting things out that are super specific, will result in a electrochemical gradient. So you will have a gradient of chemicals or ions on either side of the cell, and it will give it a membrane potential. And this membrane potential is going to be a separation of charge. Okay, so what that means is, let's take a sodium ion. It's a positive ion. We're going to live in this world, so keep an eye on these. If I have created an environment where I'm going to have high concentration of sodium on the outside of my cell, which we've talked a lot about, I'm going to have a lot of positive ions, right? If I have a lot of positive ions over here, and I only have some positive ions over here, you can see that this is going to be a much higher positively charged side than this side. So you created a high positive and a low positive. So not only have you created a high concentration of the actual molecule, you have created a concentration of the actual charge. So this is going to be a high positive, and this is going to be low positive. So there's a lot more positives outside and not a lot of positives inside. And you're going to separate it by this plasma membrane. So you've separated these charges. If you want to think about this as a battery. A battery always has a positive and a negative side. This is the same thing. Just think about a plasma membrane down the middle of it. And you are separating one charge versus another charge. The plasma membrane is acting as a battery. So that's the world we're living in right now. When we talk about voltage, the voltage is going to be the potential energy that this occurs due to the separation of the charge. So if we have a lot of positive ions on one side and not a lot of positive ions on the other side, we've created this separation of charge. There is the ability for energy to happen because we've separated these things out. That's voltage. Like I said, think about a battery with a positive and a negative side and the plasma membrane separating the two. These are measured in millivolts. So you could have an 8 millivolt battery or a 10 millivolt battery. So voltage is the potential energy that could occur due to the separation of that charge when you have different concentrations of charge in one place through another. So when we're talking about this, what we're talking about with a resting membrane potential is that your plasma membrane, your plasma membrane is actually going to separate positively, oppositely charged particles. So your plasma membrane is acting as the barrier. Here's your plasma membrane. And it is going to separate charges. And what we're doing is this voltage will only occur at the membrane surface. And this can be measured in millivolts. So it will essentially give you a charge on either side. So here is our plasma membrane. We'll talk about outside, or the ECF, and we'll talk about inside, or the ICF. This plasma membrane will have a charge. And the plasma membrane is going to be around -50 to a -100 millivolts when it is measured. So it is separating that much charge. The inside is negative compared to the outside. So the inside is negative compared to the outside. Outside. So when we say inside is negative, this could be 100. We'll get to that in a second. But the inside is negative compared to the outside. So that's something you need to know. In this resting membrane potential, RMP, resting membrane potential, there are 3 key players. There are 3 key players in this resting membrane potential. So you're going to have potassium, you're going to have sodium, and you're going to have chloride. So this is a summary. You're going to have sodium, you're going to have potassium, and you're going to have chloride. These are the 3 key players, and we're going to talk through how each player creates this plasma membrane. So we're going to start right here. How do these 3 key players keep the plasma membrane? So the first way this is going to go is you are going to have the first step, which is the diffusion of these 3 ions. These 3 ions, the diffusion of these 3 ions will imbalance the resting membrane potential and polarize the membrane. So the diffusion of the sodium, potassium, and chloride will imbalance the membrane and it will polarize it. So we're going to talk through each one individually. So the first step, the potassium is going to determine the resting membrane potential. So potassium is the key player right here. Potassium is the key player. So potassium will determine the resting membrane potential. How does that do that? So remember, after we did our sodium-potassium pump, we ended up, remember we did 3 sodiums out for every 2 potassiums in. We pumped out. So what you end up with, if we're outside the cell, we ended up with a high concentration of sodium, right? And we ended up inside with a high concentration of potassium, because we pumped 3 sodium out for every 2 potassium in. If you pump 3 sodium out, you get a high volume of sodium outside the cell. If you pump 2 potassium in, you get a high potassium inside the cell. So at the end of the sodium-potassium pump, this is what we are dealing with. We are dealing with a lot of sodium inside the cell, a lot of sodium outside the cell, and a lot of potassium inside the cell. So when we say that potassium determines the resting membrane potential, we have a lot of potassium inside the cell. So what is happening here is potassium will move out of the cell. So we will have our potassium, which is high on the inside, and he will move out of the cell. So that's what our potassium is doing. Our potassium is moving out of the cell. As it moves out of the cell, it is moving down its concentration gradient. Not only is potassium moving out of the cell, but the movement makes the inside more negative. Okay, we are moving a positive ion. If you move a positive ion out, you're losing positive ions, which means the inside of this plasma membrane, you're losing positive ions. It will become more negative because you are losing positive ions. Sound good? As you lose these positive ions, you are essentially creating a concentration gradient. So you've got potassium moving out of the cell. The movement makes the inside more negative. Then what's going to happen is the movement makes the inside more negative. Now we have a positive out and a negative in. Opposites attract. So now what we're going to have is you're going to have your potassium move back into the cell. And when the potassium moves back into the cell, it's doing that because it is a positive ion, and the positive is attracted to the negative. I know it's really weird. So first we move potassium out of the cell. When we move potassium out of the cell, it makes the cell more negative. So if we have a negative inside the cell, we have lost a bunch of positive ions. But now we have a positive out and a negative in. Potassium, which is now outside the cell, move back into the cell because the positive is attracted to the negative. So this keeps happening. The more potassium leaves, the more potassium enters. So essentially what you are doing with potassium determining the resting membrane potential is you are going to have this happen. We're going to do this whole thing right here inside the cell. So after the first step is after the sodium potassium pump. You have a high concentration of potassium inside the cell. That's what you have. After the sodium potassium pump, a high concentration in. So then the potassium will move out. So potassium moves down its concentration gradient and it will move out. As it does that, as the potassium moves out of the cell, it is causing, as the potassium moves out, it will make the inside of the cell more negative. So now we have our positive out, but we also have a high concentration of potassium outside the cell. So it's going to be positive. Inside the cell is going to be negative. What then happens is our potassium is attracted to that negative. So now your potassium is attracted to the negative. So your potassium will move into the cell from the positive to the negative. So we have a high positive of potassium inside the cell. Potassium will move out down its concentration gradient, creating a positive-negative imbalance. But now with the high concentration of potassium outside the cell, it likes to move inside the cell because it's attracted to that negative. And so what's going to occur with this process is you will have a potassium inside the cell for a potassium outside the cell. And this process will essentially continue until you have 1 potassium out for every 1 potassium in. This is how the resting membrane potential stabilizes, how potassium stabilizes the resting membrane potential, because it is moving potassium constantly across the plasma membrane, 1 potassium out for every 1 potassium in, because of this like 5-step process. When this occurs, when it's 1 potassium out for every 1 potassium in, it will set the membrane potential at around negative 90 millivolts. So due to potassium, the resting membrane potential will be negative 90 millivolts due to the movement of potassium ions only. So that's all potassium. But we also have chloride and sodium to move across the plasma membrane. So what happens with sodium? So here we go. What is happening with sodium? With sodium, remember, we're going to go way up here. After the sodium-potassium pump, we pumped 3 sodium out. So we ended up with a high concentration of sodium outside the cell. So that's where we're starting. We're going to have a high concentration of sodium outside the cell. So sodium will also contribute to the resting membrane potential. So you will have a high concentration of sodium inside the cell. That is where we're starting. So high concentration of sodium outside the cell. So sodium, how does this work? The sodium wants to move in, down its concentration gradient. So sodium wants to do this. So that's great. Sodium moves in. So if you're moving sodium inside the cell, it's a movement of a positive ion. You're moving a positive ion inside the cell. It will change the resting membrane potential. So it will make the resting membrane potential more positive. What I mean by that is we just said that potassium was the resting membrane potential negative 90. So now we're saying that inside this plasma membrane, we are at negative 90 millivolts. We are adding positive ions. If we are adding positive ions, it is becoming closer to 0. It's going to become more positive. So it's going to go from negative 90 millivolts to negative 70 millivolts. So the resting membrane potential due to sodium ions is negative 70 millivolts because we have added more positive ions inside the cell. Okay, and then the third ion we said was going to be chloride. Chloride is really easy. So it's going to be the movement of chloride in, the movement of chloride in is going to equal the movement of chloride out. So here is our cell. We're going to do chloride in for every chloride out. So that means with these guys happening, there is no effect on the resting membrane potential due to chloride. So we have sodium, we have potassium, and we have chloride. All of these are going to be involved in the resting membrane potential. Okay, so one other thing about these guys with this resting membrane potential is, why does the cell like to start with potassium? We have both high potassium and high sodium. Why does this whole entire process start with potassium for potassium to determine the resting membrane potential. The reason this occurs is the plasma membrane is more permeable to potassium than it is to sodium, meaning the plasma membrane likes potassium way more than it likes sodium. So that is why potassium will set the stage for the resting membrane potential, because it actually moves through the plasma membrane better. So this is why the potassium is the player that is key, the sodium is the player that is second, and then chloride is the player that is third. So this whole thing is how the resting membrane potential is occurring. And I know I went way past these cells, these images, but it is a very complex part. So the goal of this section is figure, I think it's 3.14. This guy right here is a visual image of everything that we just talked about, but it goes through. So active transport is going to maintain that chemical gradient, because as the resting membrane potential, you're going to have the sodium-potassium pump, and it will constantly eject 3 sodium out for every 2 back in. So you are constantly going to get this happening, 3 sodium out for every 2 potassium in. You're going to constantly get that concentration gradient. And as that happens, you're then going to have sodium pumping out of the cell will equal the rate of sodium diffusion into the cell. So as you are pumping sodium out and you're getting this at the same rate, you are doing this whole process right here. So you're going to have the plasma membrane maintained by active transport. And so this whole thing is going to occur. Not only is the sodium-potassium pump happening, but you're going to have diffusion of these ions also happening. So it's a very cool process. Okay, the last section is going to be 3.6, and it's going into the cell connecting to the environment. So in 3.6, everything we've talked about so far is cell to cell, or cell on the inside or across the cell membrane. But 3.6 is going to be the cell interaction, cell to the environment interaction. So we're going to go through these real quick. So how do we get information from the cell into information inside the cell? So cells will interact with their environment by either responding directly to other cells or to chemicals. And we're going to bring up the concept we've already talked about, which is going to be the cell molecules. So the CAMs, the cell sticking to other molecules, or the receptors that are on the plasma membrane that can communicate to other cells. So just kind of a brief overview of cell adhesion molecules. Every cell has a bunch of these really, really sticky proteins. So this is common on every cell. Some general reasons why you have these sticky proteins is it will anchor the cell to another cell. It will cause the cells to move past each other. It's really important with the immune system and white blood cells. And it can actually stimulate the synthesis or degradation of adhesive membrane junctions, essentially meaning it can create cells to stick together better. It can create new junctions or some sort of signal. When we're talking about these plasma membrane receptors and these receptor proteins and these signaling, what I really want you to know is that in the world of signaling, this is the concept really for this section. Chemical signaling, it's the interaction between the receptor and the ligand that can change cellular activities. So what that means is if I have my plasma membrane of one cell and my plasma membrane of another cell, and here is my receptor protein, and here is my receptor protein, whatever signal is on this cell will trigger a signal here. And that will trigger something to happen inside the cell. So it will actually trigger some sort of process inside the cell, all because it picked up some sort of communication outside the cell. So we talked about this when we were talking about cell-to-cell interactions. It is the exact same thing when we're talking about cell to environment. So it is the same thing when we talked about cell-to-cell interactions. Your plasma membrane of the cell that is getting communicated with, you're going to have your protein, and you're going to have some sort of environment in the cell. So what this is happening with the cell-to-cell interactions or the cell-to-cell communication, the environment usually going to be a hormone, it could be a neurotransmitter, will connect with the cell, and that will cause some sort of process inside the cell. So jumping to this figure right here, you do not need to memorize this or know the sequence of steps, but I want to give you a heads up. So right here, what we're looking at is here is the communication outside the cell, and then this is going to be the communication inside the cell, and here is that protein we were talking about. So this is a very complex, this is actually called a second messenger system, but what I want you to see here is all we're saying right now is that there's some sort of chemical on the outside that will bind to this protein, and it will essentially create some sort of process inside the cell. However, what you need to look at here is we have one step, we have two steps that cause something to happen inside the cell, we have three steps that cause something to happen inside the cell, we have four steps that cause something to happen, five, and then finally the actual process. So it is not as simple all the time as one connection, then communication, it is not always as simple as here's the connection, the communication, and then we cause one process. Sometimes this can be 6+ steps that are essentially going to then create some sort of process. That is how biochem and physiology is really going to play, is knowing not only the signal here, but also to get this output of some sort of output process here. It's going through every single step along the way to get there and knowing why every step occurs and what can go wrong at every step. So it's not just as simple as one process inside. This is really the complex of how this class is going to be organized with the sequence of steps and what can go wrong along each one. So this is going to be Chapter 3, Part 1, 3.1 to 3.6.